Nee.
Hello, everyone. So if I was asking you to imagine polar regions for me, you might think of something like this top left image here. And whilst being very pretty, there's arguably not much biomass, at least on first glance. But polar regions are actually highly productive, and phytoplankton blooms, like this group here, are of high interest to marine scientists like myself. Phytoplankton form the base of the food web in marine oceans, and this impacts ecosystem functioning as a whole, but also things like carbon export and nutrient cycling, which is also relevant for ecosystem services and in the light of climate change. However, if we want to look at how these microbial communities are functioning, who is there, what are they doing, we're still mostly relying on traditional microscopy. And traditional microscopy is tedious, to say the least. It's slow, it requires extensive expertise, and it's also still very prone to human bias. So our solution is to use a so-called multispectral imaging flow cytometer. The Amnis image stream is able to take images where we have both traditional community composition information, like on the left here, but also functional biodiversity through the use of fluorescent stains. So we are able to get for every cell that passes through the instrument, we are able to get both bright field images and fluorescent images, as well as hundreds of features which are calculated through the use of embedded masks. Obviously, this gives us a huge data set, and therefore, we would like to apply deep learning to help us in tagging these data sets. So PolarFlow is interested in using deep learning to figure out species identification, who is there, and through the use of staining extent, what they are actually doing in that ecosystem. And our vision is to turn this into, and to grow this into large-scale functional biodiversity monitoring, both in polar oceans and beyond. So if this is a use case that sounds interesting to you, if you have overlapping data, or if you also have similar images, I would love to speak to you in the poster session this afternoon. My poster number is 1375, and I look forward to talking. Thank you.
Thank you. Our next speaker is Wang from the Friedrich-Alexander-Universität in Erlangen-Nürnberg.
Yeah, hello everybody. And today I will present my SoraCT, and this is an unconditioned 3D CT generation model, and the inspiration is pretty simple. One day I use slicer to check the slices, CT slices from the neck to the abdomen, and I found when I move the button, it's totally a continuous video. So why don't we try a video generation model to do the CT generation? So at that time, I just asked myself, who is the most effective video generation model? Then, of course, Sora. And I use Open Sora for this project. It's not the real Sora from OpenAI. It's just an open source Sora. And it uses MMDIT to do the generation things. And you can see here, I use CT rate dataset from UZH, and I just cut the CT2 slices along the z-axis. And you can see here is our pipeline here. Preparing the data set is really important because nowadays all the things is about data. And we use different resolution sampler to have all the different resolution data. And we just use the Sora video generation to train those videos, to train those slices. And we generate reasonable CT here. You can see here. And And you can see our results, evaluation results there. And our FID is lower. That means our image quality is higher, and our FVD also lower. That means our image is more continuous along the z-axis. And you can see and the last one is the time of generation. And you can see we can save a lot of time during the generation phase. And what you can do with our model, I think is a really basic model. You can use our model to do the second step, like text to video, text to CT generation, or you can use this to train a new segmentation model by RGB generation mask, something like that. Yeah, and that's all my presentation, and thank you.
Thank you. Next up is Kilio from the Institute of Stroke and Dementia Research in the LMU Clinic.
Thank you. Shall we get started? OK. Hello, everyone. My name is Kilio, a PhD student from LMU Munich. Today, I'm happy to be presenting our work on a 3D foundation model for generalizable biological structure segmentation in tissue clearing images. Help? How to flip? OK. OK. As we know, the CAR-T therapy is a major breakthrough in immune therapy, but these still are extremely rare and carrying weak fluorescent signal and show dynamic infiltration behavior, which making the detection highly challenging. So we are facing this several problems. First, localizing the CAR-T cell within whole tissue, and simultaneously detecting single-cell cancer metastasis, and additionally enabling scalable high-throughput quantification across different data sets. Here is our contribution. As we can see, to address this challenge, we built an end-to-end AI foundation model with 3 modular. One organ modular for whole body organ segmentation, one cell modular for both CAR-T and tumor cell detection, and lastly, quantification modular for large-scale co-localization and longitudinal analytes. Let's move to the experimental side. This shows how we got our data. We injected the CAR-T cells and the leukemia cell into mice, and followed by the nanoparticle perfusion, V-disco tissue clearing, and light sheet microscopy. We got unbiased whole body scanning. This slide shows our computational pipeline. Generally, it has 2 parts in parallel. The first part is our organ segmentation mask generation, and the second part is CAR-T tumor signal quantification. Regarding the model training and performance, we annotated our training data using VR and evaluate by voxel White's dice score. And for the benchmarking, we compare with several methods and achieved the SOTA. This shows the whole body results of the inference. As we can see, we inference the whole body organs and then evaluate some ROIs, like the whole lung. And zoom in, we can see the single-cell resolution spatial distribution among the whole tissue. What's more, we also did the longitudinal quantification across many mice. And we found that our model has zero-shot generalization on complete independent data sets as well. And more funding should be introduced in our poster. And thank you for your listening.